for everyone coming and sticking the last one last grade. So my name is Sean from Texas A&M, and today I'm going to show you how can we use the genomic data to study population genomics and how this information can help us with conservation down the road in beetles. So why beetles? How do you ask me? Evolutionary biology often say God has an importance for beetles, because beetles make up a quarter of biodiversity. Ever since when I was little, I've been amazed by how diverse they are. Some of them are colorful, some of them have a huge horn or pincers. So this curiosity led me to study their evolutionary and vertebrate history, especially in species. They are endangered and endemic because a lot of time they are suffering from habitat fragmentation. So I want to protect them, so the future generation can see them and appreciate them. So habitat fragmentation can happen a lot of reasons. Sometimes due to the natural event, like a river, or a lot of time it's caused by human activity. As a result, habitat got broken up, population gets smaller and more isolated, and gene flow between them got cut off. So when population got smaller like this, become more valuable, like things like genetic drift. The genetic drift will reduce the genetic diversity. So the loss of genetic diversity can make it really hard for those population to adapt to the change in the environment. So what's genetic drift? Just give you a quick review. Genetic drift is allele frequency change just by random chance. So there's no direction. So image you have 100 balls and they are 4 colors with the same frequency. I ask you to take 10 balls with replacement for 10 times. You take 10 and put it back for 10 times. Just by random chance, you'll change the frequency. I ask you to do it again. By random chance, you'll change the frequency. So this time, you probably noticed that blue ball are gone. Here's a simulation I make. In the left-hand side, it's a smaller population. And the right-hand side, it's a bigger population. So you can see the smaller population, the allele frequency fluctuates a lot. When you go all the way up, we call fixation. All the way down, we lost the allele. So today's talk, I'm going to focus on 2 parts. The first part, I'm going to talk about the genome assembly. The second part is going to talk about the population genetics, how we use the genome assembly to utilize the genetic data. So the first cool beetle I want to introduce to you is a long-arm beetle, Cheratonis. So this is a genus in scarab beetle. And there are 10 species, and they are all in Asia, and 3 of them are endangered species. So the main threat to this beetle, the first is habitat loss, because the main habitat for them is highland forest. You can see the gray area over there because of climate change and anthropogenic activity. And the second thing is the second one is illegal collection, because they are the biggest beetle in Asia, and also the male have an exaggerated long arm, and also their color is metallic green, so everyone to collect them, including me. We do have permit. For this project, I want to assemble the chromosome level genome assembly for the long-arm beetle. I choose Cheratonis formosanus, which is the Taiwanese endangered endemic species, and where I'm from. So for the genome assembly, I'll use a hi-fi PacBio platform. I'll sequence the male, so I can try to get the Y chromosome. But I also will use a short read to sequence the female, so I can try to identify the sex chromosome. I'll also use the Hi-C data, try to scaffold to the chromosome level. And the last one, I will use the RNA data to help the genome mutation. I don't have much time, so I'm just going to show you some results. So usually when I look at the genome assembly, there are 2 things we look to see if this is a good genome assembly. The first thing is contiguity. So it's how continuous the genome is. The second thing is completeness. So it's how much genome that we really actually capture. So the plot I'm showing you here is I call a scaffold coverage plot. So the way we read this plot is each bar you can think about it's a scaffold. So first we look at the largest scaffold over here. So this largest scaffold is around 93.3 megabase, which take about 30% of the genome. And then we go to the second largest and third largest until we cover 100% of the genome. So here I'm just going to show you some state of this genome assembly. The genome size is around 600 megabase. With 62 scaffold, and N50 is around 83.3. So this looks pretty good. The second thing is the completeness. So I use the Busco benchmark universal single copy ortholog. So we believe those genes were present in in this clay. So the Busco score is around 99.3 with 0.9% duplication. If you work with the genome assembly, this is an amazing result. And also this is a non-model. Here I'm showing the high-C heat map. So the high-C heat map is showing you the 3D organization of the genome by displaying how often the different genome, the chromosomes interact with each other. So the stronger the interaction, the more intense signal. So you can see there are main 10 scaffold. But beyond that, high-C data also can help us to identify the misassembly. Like I mentioned before, we want our assemblies high contiguous, right? But a lot of times the assembler will misassemble. They put two chromosomes together. But with the high-C data, we can spot that and we can break them down if necessary. And this is a synergy plot. So it just shows the genomic similarity and structural differences between two genomes. So on the top, I have the long-arm beetle I assembled. On the bottom, I have the Japanese rhinoceros beetle. They published on NCBI. So you can see the scaffold, they are well connected, which means our genome assembly is very consistent with close related species, which is a good sign. So in the inner ring over here, you are looking at the normalized read depth, so range is 0 to 1. So I mapped the female read, short read, back to the genome I assembled. So you can see 0 to 1 there read depth. But I want you to draw your attention, look at the scaffold 10 or chromosome 10. Don't blink. Look, so I mapped the male back to the genome. So don't blink. So you see, because the male, male only have one copy of the X chromosome, right? So you only see a half copy. I hope you capture that. So I'm gonna switch the gear. So right now we have the genome assembly. How can we utilize this genome assembly to infer population genetics? So second beetle I want to use the Western Hercules beetle by Nancy Grantine. This is the largest beetle in North America, and that's my hand as a references. And so they're endemic to New Mexico, Utah, and Arizona. So when you think about these places, about the habitat, what do you picture? Desert, right? So this is me in the Scarlet National Park. But actually, there's a lot of tree cover over there. So in this area, we call Mangolan Ring. So Mangolan Ring is a really big geographical structure stretched from northern New Mexico to northern Arizona. So actually the tree over there, it's pine oak or pine oak mix or pine forest. But I want to draw your attention to look at this area we call Sky Island. Sky because it's really high. I'm talking about 7,000 to 10,000 feet high. Island because they are isolated by the desert. So I'm really interested in the population. So this is a distribution of dynastic grantine. So I'm wondering if they do have gene flow and especially those little population, which I really care about. So in order to study this, I need to collect a specimen, from Mangolan Ring, also with the Utah and Sky area. Okay, so those are my study side, and so the first step is a sample collection, and I really appreciate my collaborator JP. They got a specimen for the Mangolan Ring in Utah from the Field Museum and sequenced that. And in the past 2 years, I went to the Sky Island, just collect all the beetle and bring back to my lab. And so I did all the DNA extraction and sent out for the sequencing. And for the data analysis, the first step is called mapping. So JP, Dr. Huang's lab, they assembled the Hercules beetle genome. It's published last year. And so I use that reference genome, map all the short read back to the genome. And then I can do variant calling, and you'll get a VCF file. And for this file, you can do a lot of downstream analysis. And so here I'm gonna just show you some analysis I did. So the first thing I want to understand the population structure. So I start with the PCA using the print. And so this is a non-model based method. So from this plot, you can see there are 4 distinct clusters. So one from Mangolan Ring, one from Utah, and one from all the Sky Island, and another one from Mexico and some unknown location. That's just, we purchased the specimen, but they told me it's from here, but DNA don't lie, you know? Mexico, so I said, yeah, we cannot say that. So you can see for cluster, and so this is not model-based. And the next one, I used a plot cluster. So plot cluster, this is the model-based approach. So this is a mixture plot. And you can see over here, the k equal to 4 is the the best fit, which match what we saw in the PCA. What is kind of surprised is that we see all the Sky Island, actually they are in the same population structure, because I also work with different beagle. Actually, the finance sprint, actually they have a very impressive flyover dispersal ability even across those isolated mountains. The next one, what we can do is show the demographic history. So this is the MSMC plot, and show the effective population size have changed over time across 4 different regions. We can see all population were during the Pleistocene, they have a really high effective population size. This could be they are connected, or they do a gene flow. But after the Holocene, they are kind of like separated. And the Mongolian repopulation seems to have remained very large and very stable. But individual from Mexico or Utah population have gone through sharper decline, suggesting that they have more isolated or experiencing some strong genetic drift or bottlenecks. And so I hope this this short talk and this workflow I can convince you that the long-read sequencing has make genome sampling more accessible especially for non-model organisms. And the reference genome is very essential for population genomics because it provides foundation for understanding genetic diversity and evolution. And the last is future direction for the Hercules Beetle project. I want to investigate the signal for selection across the population, particularly those smaller populations, if they do have local adaptation or selection processes. And this is just another beetle I'm going to work on. This beetle is really rare in the US. You only can find in White County. So right now I'm going to sample this genome and also going to extract all the short-read RNA, short-read DNA. And with that, I would like to thank all my lab, my PI, Whiteman, and all my lab member, and especially my collaborator, JP, Dr. Wang, providing permit and sample for me. And the last part is time for a quick pitch. I'm looking for a postdoc next year. I'm especially interested in genomics and PopGen, population modeling. And that's my website and email. And what I can bring to the table is the first, field work. I do a lot of intense field work. I'm from Texas. I drive to Florida, Arizona by myself, and even here, I do everything. And I do all the wet lab. I can do DNA extraction, RNA extraction, high molecular weight. I can do everything. Bioinformatics, I process all the short read and everything till analysis, so you don't need to worry. So if you're a postdoc, somebody doing postdoc, please let me know and connect with me. And with that, I'd like to take any questions you may have. Thank you.